In this chapter, we will first discuss the tertiary structure of DNA, which, as you recall, is complex packing arrangements of double-stranded DNA in chromosomes. So what we will talk about is chromosome structure. We will also discuss or examine DNA found in cytoplasmic organelles. We will mostly focus on mitochondrial DNA. Now, before we start look at the chromosome structure, I want to make 2 points. First, the length of genetic material far exceeds the length of the cells. Therefore, each cell has to come up with mechanism how to tightly fold and pack this genetic material in the provided space within cellular nucleus or within cell in bacteria. Second point I want to make here is if this DNA is so tightly packaged, it is not accessible to, let's say, transcription factor or enzymatic machinery which replicate DNA. So in order for DNA to be replicated or transcribed, it has to unfold. So cell had to evolve mechanisms how to actually switch DNA from tightly fold to unfold and how it refold back. Now, look at the example book. You try to read a chapter from book, but book is closed. So the pages are so tightly packed in closed book that you cannot see the letter, so you cannot read it. In order for you to read, you have to open book, and now you can see the letter. The same apply for the DNA replication or transcription. Folded DNA has to be open in order that the replication or transcription machinery can access it. Okay, so now let's proceed, and first what we will talk first about will be supercoiling, which is an important tertiary structure of DNA which helps DNA to fold and package in the space. So as said before, the length of genetic material far exceeds the length of the cell. Now, some example, simple E. coli, our good friend living in our guts and providing us with many benefits, has a genome length approximately 4.5-4.6 million base pair. If you stretch these base pairs, this DNA would be about 1,000 times longer than bacterial E. coli cells. So there must be some packaging problem here, or respectively bacteria had to evolve certain mechanism how to package this DNA into the cell. It is even this phenomenon is even more pronounced in eukaryotic human cells, where we have more than 6 billion base pair of DNA, which if you stretch can measure somewhere between 1.8 to 2 meters, which is 14,000 times more than the length of the cellular nucleus. So clearly DNA molecule must tightly packed and into this small space. And what is actually mechanism? The first mechanism which we will discuss is supercoiling. Supercoiling can be positive or negative. So we start with relaxed state. So the startup point to define what is over or under rotation is if you start with relaxed state, which is the lowest energy state for bDNA, and which is, you remember, approximately 10 base pair per 1 turn. So if DNA has approximately 10 base pair per 1 turn, it will be relaxed. Now, relaxed DNA, also low energy stage, is not the optimal for packaging into the nucleus or into the cells. So in order to bring higher level of organization, DNA is supercoiled. It is either over-rotated if DNA is over-rotated, we say that it's positively supercoiled, or it can be under-rotated, which means that it's negatively supercoiled. So how the coiling changes in DNA? There are specific enzymes, topoisomerase 1 and 2, which are responsible for adding and removing turns in DNA. They can cut one strand or both strands depending which topoisomerase is present, and they can add, rotate strand around each other and add or remove rotations. It might be hard to imagine supercoiling. If you struggle with this concept, I advise you try to find an old telephone. Maybe you have it somewhere in back drawers still stored. So if you find it, take a telephone cord and try to look at it. If it is relaxed, this is how it looks when it relaxes. It resembles DNA when DNA is relaxed, when it has 10 base pair per turn. Now, take that cord and try to bring more rotation. Try to over-rotate and fold it on the side. What you will see that it would actually fold on each other. This cord would supercoil. This is what supercoiling is. What is interesting is that also when you under-rotate it, you bring the cord to a relaxed state and you under-rotate it, you start to remove on both end rotation and you fold it, you will again get supercoil. However, what we say that if you under-rotate the DNA or the cord, we have negative supercoiling. If you over-rotate, you add more rotations, you will get positive supercoiling. In cells, the most DNA are negatively supercoiled. Why it is? Supercoiling is important for assuring the DNA takes less space that it can fit into the space provided. However, negatively supercoiled which is under-rotated would separate easier than over-rotated DNA. So cell would need less energy to unwind under-rotated or negatively supercoiled DNA than it would do if the DNA was over-rotated. So remember, DNA has to be unfolded in order to replicate and transcribe. So that's why the predominant form in cells is under-rotated, negatively supercoiled DNA. Now, if you still struggle with understanding positive and negative supercoiling, let's go to next slide. So I hope that this slide which I draw would help you to understand a little better supercoiling. So first, let's look at a relaxed DNA. You see that one turn in relaxed DNA, you remember B form has 10 base pair per turn. So here are the outline, here are 10 base pair per turn. Now, if you introduce more rotation, if you positively supercoil DNA, which is outlined in red, so look here, so one turn will have less than 10 base pair per turn, because those 10 base pairs you had here were now divided between two turns. If you have negatively supercoiled DNA, you will have more than 10 base pair per turn. In negatively supercoiled, you see one coil goes over right here, so you have 10 and plus additional base pairs. So the number of base pair per turn change according stage of supercoiling. Let's do some example. Let's say you have a DNA which has 100 base pair to make it easy. So what would be relaxed DNA? So relaxed DNA would have 10 rotations because there is in relaxed DNA there is 10 base pair approximately per rotation. What about negative supercoiled? So negatively supercoiled DNA would have less than 10 rotations. So it can have 9, 8, 7 or less rotations. So if you divide, let's say, 100 base pair by, let's say this DNA has 8 rotation, you immediately see that you will have more than 10 base pair per turn. If you have positively supercoiled DNA, so you introduce more rotations than 10. So here I want to point at the number of 10. Here, this is a base you take when you compare two, when you look at the negatively and positively supercoiled. So if you have positively supercoiled DNA, so you will introduce more than 10 rotation. So it would be 11, 12 or so on. So more than 10 rotations. So what it will give you? So you will have per one rotation less than 10 base pair, because now those 100 base pairs will be divided to 11, 12, 13 or whatever rotation you will introduce. So I hope that supercoiling now is clear. So let's proceed and discuss the structure of the chromosomes. First, we start with bacterial chromosomes. Most bacterial genomes consist of single circular DNA. Also, I have to point that there are some exceptions, and you can find some bacteria which possess linear DNA, but predominantly bacteria possess one single circular DNA molecule. This DNA molecule is actually looped, and if you open bacterial cells, you will see something like this outlined in the picture, and you see that the DNA forms the loops. What was suggested that these loops actually are held together, are complex with proteins, which actually holds the bacterial chromosome in looped in supercoiled organized space or a shape. Be aware, bacteria do not have histones. These proteins are not histones. You find histones only in eukaryotic DNA. These proteins are very simple protein which actually associate with bacterial DNA, but they are not histone. Now, if you draw bacterial cells, you know that bacteria are prokaryotes, so they do not have nucleus. However, the DNA usually clamps. You find it in discrete place where it forms nucleoid. Okay, so it's not nucleus, it's nucleoid. In addition, bacteria can possess independent double-stranded DNA, so-called plasmid DNA. And we will talk a little bit more about usage of plasmid in molecular genetics. We already discussed plasmid in chapter 9 when we talk about the bacterial and viral genetics. So, as you recall, the plasmid can replicate or replicates independently of bacterial chromosome. Similarly to bacterial chromosome, eukaryotic chromosomes consist of a single extremely long molecule of DNA. So you can imagine that even during interphase, this long molecule of DNA has to be condensed. It is highly condensed. It undergoes further extreme condensation during cell cycle. This extreme condensation is necessary for chromosome movement in mitosis and meiosis. So this change in chromatin structure from highly condensed to extreme condensation is necessary. So as you can see, the chromosome structure is not static, it's highly dynamic, and it changes during cell cycle, and it also has to change during DNA replication and gene expression. Now, what is chromatin? Chromatin refers to DNA plus protein. Eukaryotic DNA is closely associated with protein, and the structure where you have DNA and protein is called chromatin. And there are two basic kinds of chromatin, euchromatin and heterochromatin. Euchromatin is one which undergoes normal cycle of condensation and decondensation during cell cycle. It's a chromatin. Euchromatin is one where you find most of gene which encodes protein which are expressed during certain time of the cell cycle or cell life. Now, what is heterochromatin? Heterochromatin remains in highly condensed state throughout the cell cycle, so it doesn't change during mitosis, meiosis, or interphase. Now, where do you find it? You find it in each chromosome, actually the area of centromere and telomere, highly repetitive sequences which do not encode genes, or there are genes which stays in heterochromatic stage. You will find very few genes in heterochromatic regions, and generally there is lack of transcription crossing over, and heterochromatin does not replicate, DNA of heterochromatin does not replicate at the same time as euchromatin. It is usually delayed, and it takes place at the end of S phase. Now, heterochromatin can be either constitutive or facultative. Constitutive is one which you find, for example, in centromere and telomere. It's a permanent heterochromatin. Facultative heterochromatin is one example of it is X chromosomes. It's heterochromatin which occurs during certain developmental stage, and we discussed already X chromosome inactivation. As discussed before, eukaryotic DNA is closely associated with proteins, and the most abundant proteins in chromatin are histones. Histones have a high content of positively charged amino acid, particularly arginine and lysine. These positive charges in histone attract negative charged DNA. You remember phosphate of DNA has negative charge, and this attraction between positively charged histones and negatively charged DNA holds the DNA in contact with histones. Now, there are different group of histone H1 and histone H2A, 2B, 3, and 4. The histones, the later 4 one, form the octamer within the nucleosome, which actually around which the DNA wraps and forms a nucleosome. H1 histone usually seals the nucleosome and keep it stable. Now, beside histones, there are additional non-histone chromosomal proteins, such as scaffold proteins, proteins of replication machinery, proteins which are necessary for gene expression, kinetochore. We talked about kinetochore in chapter 2 already. Now, before we go to the structure of the chromosome, nucleosome, and chromatosome, I would like to point at one fact: histone modification. We will talk about histone modification later on in epigenetic. However, histone normally, through their positive charge, tightly interact with DNA and assures this tight interaction and packaging. Now, this interaction has to be released during gene expression, and this release is mediated through histone modification, through histone acetylation, so through epigenetic changes. There are also changes within histone, which might be methylation. There might be also changes in DNA, such as DNA methylation. As you recall, DNA methylation affects stage of gene expression. So we will talk later on during the course about epigenetic and epigenetic changes. As we mentioned before, DNA plus proteins form chromatin. Chromatin has highly complex structure with several levels of organization. The fundamental repeat of the chromatin are nucleosomes. You can see them right here. Nucleosomes pack together and form 30 nanometer fiber. We will talk a little bit later about that. What I want you to realize is the stage of condensation and the shape of histones. So when you isolate chromatin from the nucleus, it frequently looks like beads on the string. right here. You see little beads on the strings. So those beads are nucleosomes, which are formed by octamer of the histones. You remember histone 2, H2A histone, 2H2B histone, 2H3 and 2H4 histones form this core histone or octamer around which the DNA wrap. Now, in between of this histone, core histone, in between of nucleosome, you have linker DNA. Now, if the DNA is in extreme condensation stage, linker DNA is not visible or is not accessible. How can you distinguish that? If you treat DNA with nucleases, if you treat DNA with DNase, if the DNA is in only condensed stage, not extreme condensed, it would cleave right around, nucleases would cleave right in the linker DNA sequence because the linker is not packed, is not associated with histones. While if you have extremely condensed chromatin, this linker DNA is not accessible, so DNases will not work. Now, what I want to point at here also is that in the histone octamer, each histone has a flexible tail which contains positively charged amino acid, and they interact not only with nucleic acid, but they can also manage interaction or mediate interaction between nucleosomes. So let's now review chromatin structure. We already talked about the primary and secondary structure of DNA, so let's talk about tertiary structure. So the repeated unit of tertiary structure is nucleosome. Nucleosome is formed by the octamer of histones, and DNA is wrapped around this octamer. So nucleosome is DNA wrapped around the octamer of histones. The octamer is formed by two H2A histones, by two H2B histones, two H3 histones, and two H4 histones. So higher level of the organization after nucleosome is chromatosome. What is chromatosome? Chromatosome is nucleosome which is stabilized by histone H1. So each chromatosome has octamer DNA wrapped around it and one H1 histone. So H1 histone interacts with DNA at the site where it enters and exits nucleosome and kind of stabilize chromatosomes. Chromatosomes folds up and produce 30 nanometer fiber. They actually supercoil in zigzag manner and forms this 30 nanometer fiber, which further can form loops of 300 nanometers width or length. So these loops and actually even 30 nanometer fiber are stabilized by protein which associate with chromatin. 300 nanometer loops are packaged and fold in producing 200 nanometer fiber. You see they are packaged here and produce 200 nanometer white fiber. Now this 200 nanometer fiber in turn produces highly supercoiled structure, helical coiled structure which has 700 nanometer width, and this is what you see as a chromatid, one chromatid. So each chromosome you can observe in metaphase, in mitosis, has one chromatid which has 700 nanometer width and second chromatid which has also 700 nanometer width. So you can see that is extremely condensed chromatin which you can see during cellular reproduction during metaphase of the mitosis or meiosis.